The scientist's mind is wired to probe nature and discover the curious ways the interactions within its world occur, but oftentimes does so without considering the potential consequences of a discovery, its implications and possible implementations, whether for the improvement of mankind or against it. When Jennifer Doudna, a biochemistry professor at UC Berkeley, was studying bacterial and viral immune systems with her team, this was her exact state of mind. Together, they discovered DNA strands in bacteria and viruses called CRISPR that, with the help of the protein of Cas9, detect any virus or bacteria and essentially shred it by unzipping its DNA. Describing this in her findings in her book, A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution, Doudna emphasizes that this process evolved in bacteria and viruses as a means of survival and has been perfected through Darwinism. When Doudna and her team first discovered this, their first thought was to publish it and make it available to the scientific community to discover more about it. This also had an unintended consequence. Companies began to use this in labs to alter human body cells with the CRISPR and Cas9, known as somatic cells, in order to heal certain diseases, such as HIV. CRISPR was extremely effective in doing so, some experiments fixing 99% of mutated genes, unlike previous methods that fixed 30% of them in the first trials. Furthermore, it was cheaper and could be programmed to attack any mutation containing any number of nucleotides, the letters making up DNA, not just a specific number, making its potential boundless. Doudna separates her book into two different parts. In the first part, she describes CRISPR, how it works, and how it was discovered, while in the second part, she describes its possible uses and then describes her view on what should be done for now. Let us first look at the system of CRISPR that Doudna examined while looking at the immune systems of bacterial cells and how it works. Doudna first identified CRISPR and its function in a bacterial cell when looking at its DNA. The bacterial DNA had repeated sequences of the same nucleotides, which have been called by previous scientists CRISPR, standing for clustered, regularly interspaced, short palindromic repeats. In between these sequences of DNA stand other nucleotides that code for what were called Cas9 proteins, and Doudna was the first to discover what these did. The system is made up of two parts, the protein itself and a guide RNA. Together, they are called the Cas9 complex, which can identify and cut DNA of any length with accuracy and precision because of the RNA. The guide RNA is programmed to identify a spot along a DNA strand to cut, a PAM. The RNA, made up of a single strand of nucleotides, unwinds the double helix of the targeted DNA at that spot by binding with one of the two strands of the DNA, with whose nucleotides it matches. This then enables the Cas9 protein to cut the DNA, which scientists have previously called a double-strand break. The cell will then proceed to repair the break by forming new nucleotides at that spot, but this process makes many errors, since it just cares about filling the gap and not replacing the missing nucleotides with the same ones. This leads to the inactivation of the gene. This is what Doudna discovered, that bacterial cells use this to inactivate the genes of viral attackers that would try to incorporate their DNA into the bacterial DNA. Now, researchers have discovered that they can deactivate Cas9's cutting enzyme and instead add new different enzymes that can change nucleotides. This makes it so when the RNA unravels the DNA, the new enzymes can change the targeted DNA accurately and precisely. This can be used to fix DNA mutations at specific spots for any DNA. This can also be used to introduce different codons, such as a stop codon, which can stop an unhealthy version of a gene from working and from further ones being made. Cas9 is already an extremely powerful tool, but researchers have yet to exploit all of its capabilities. To develop her explanation of CRISPR in the first part of her book, Doudna first describes the general principle that gene editing is based on, which is that vessels are used to carry new DNA into the nucleus of cells. Not how effective one method of it is depends on how well it can incorporate the DNA into the actual genes of the nucleus. She then recounts the various uses of gene editing that have been used in the past, including ZFNs and talins, and how they were discovered. She weaves these into her own past, describing where she was at the time that these discoveries were made, whether it was in graduate school at Harvard, working on her PhD involving the shape of RNA, in Boulder as a postdoctoral researcher, or whether it was in her undergrad years. She then chronologically recounts her research in the years before she started working with CRISPR, and then describes how she first heard of the term and was then able to find out how it works. By listing things in the order that they happened, it is easy to follow what happened and when it happened, and the reader can clearly see the development of the research behind CRISPR through time. However, in doing so, Doudna describes the experiments that her team carried out in great detail, which can be hard to follow at times, especially because her team includes members from across the world, such as Emmanuel Charpentier in Berlin and Christophe in Poland. They are all carrying out experiments, and Doudna describes them with the correct vocabulary, not layman terms, such as cell electrophoresis, bacteriophages, and nucleoses, and instead defines the correct vocabulary and explains it. Personally, I enjoyed this, but this made the style more textbook-like at times than narrative-like, which some people may dislike. This may also hurt Doudna's purpose, which is to spread the knowledge of CRISPR to the general public. Since this book was so hard to understand at times, some of the general public might start reading this book and not want to finish it because half of the book is describing these CRISPR using these terms in this vocabulary in great detail. In the first part, Doudna sprinkles in different anecdotes from past experiences, whether it be how her love for science came from her curiosity during her childhood with Hawaiian wildlife or her first time in a professional research lab, in order to qualify science and its community of researchers. These work extremely well and are intertwined with her research, the assertions making clear what motivates her and the people involved with her research during the time. She also sprinkles in various diagrams in the form of sketches to help the reader picture what is actually happening in DNA, since she explains it in so much detail that it can sometimes be hard to know exactly what is going on. These diagrams are truly extremely helpful, especially since the vocabulary she uses is so plentiful that it's hard to remember it all. Let us now focus on Doudna's second part of her book. During the second part of her book, Doudna focuses on the implications of her team's discovery, the consequences she had not been thinking about while doing her research. To do this, she uses many examples of hypothetical situations, applying CRISPR to current problems. She focuses on what CRISPR could mean for animals, specifically livestock and plants. Addressing the topic of GMOs, Doudna believes that the criticism it has received is unwarranted, and its negative connotation is due to a small handful of studies that claim to reveal adverse effects on consumer health or the environment, even though they have been discounted by the broader scientific community. Looking at the spread of disease, she describes the fact that CRISPR could be used in various cases to save an uncountable amount of lives, such as making mosquitoes resistant to malaria in certain parts of Africa. Furthermore, she argues that one day, pigs could be used to grow certain human organs inside their bodies that could then be used for human transplants, and that various animals such as cows could be made fatter and more muscular to increase food production by inactivating the gene myostatin. This already occurs naturally in rare cases, and since this gene also exists in humans, 2 cases of its inactivation have been reported, one in a Michigan family and one in a child in Berlin, the condition known as muscle hypertrophy, or double muscling. This helps Doudna transition to her next point, that CRISPR could be used to genetically alter humans to improve them, and make those changes permanent by introducing them in the germline, which are the gametes in the ovaries, so that any descendant inherits the changes as well. Doudna tells us that germline editing could provide a one and done curative treatment for patients, which is why various countries prohibit it, including 15 of 22 nations in Europe. For this reason, Doudna believes a worldwide moratorium, a temporary prohibition, should be held of germline editing for now. CRISPR, being extremely effective as it has corrected 99% of genes correctly in some experiments, has still been inaccurate in 1% of the genes during trials. At times, it cuts the wrong genes out of the DNA, which could be essential genes to the function of a cell. Until this is solved, germline editing should absolutely not be engaged in, but even there are more issues. As Doudna affirms, germline editing may be used to increase the frequency of genes that are beneficial in one generation, yet the same genes may be harmful to future generations. This is due to the fact that some genes provide protection against certain diseases, but increase susceptibility to others, such as a variant of the dark gene that provides protection against malaria, but also disposes people to be more susceptible to HIV. I completely agree with Doudna. Nature has perfected this process of transferring DNA for millions of years, and still makes mistakes as seen by cancers and other inherited mutations. After we humans have discovered this process to alter genes a mere 4 years ago, we should not be using it to alter our germline. which could lead to many unintended consequences that would be irreversible. Furthermore, Doudna argues that since in most communities around the world, there is already a great disparity between the poor and the rich, creating a genetic disparity as well, where the rich would clearly have access to more gene enhancement than the poor, would lead to some groups being severely disadvantaged from birth. This clearly makes sense, and before any human is allowed to undergo genetic changes in this fashion, there needs to be a large discussion across the broad public, not just the scientific community, since the effects of it would ripple across the world. Governments need to be involved in the policy discourse in order to establish clear rules and laws that prevent this from happening, and to define clearly what enhancement is. Scientists have a difficult time deciding what is considered enhancing, since there is no difference genetically between altering one DNA strand to make someone have more muscular mass than normal and altering another DNA strand to remove HIV or a cancer from one's genome. To decide how to limit gene editing and how to define enhancement is a necessary discourse for everyone to partake in, because it will not only affect the scientific community, but the whole human race. Not just scientists, but any regular member of the community may be enhanced. To emphasize her point that discussion must be an undertaking before any action, Doudna is self-effacing despite having fully explored CRISPR. She is unassuming and modest, affirming she does not have all the answers, and that many issues associated with CRISPR still need to be explored. Realizing one's limits and involving others to join in the discussion to hear diversity of opinions and views conveys intelligence, a fantastic way to reach a solution and compromise. Many people choose not to hear others' opinions and believe they are right or have all the answers, but her smarter action is to hear all the different perspectives. Furthermore, she is honest with her audience, describing her doubts about what she had done about her discovery and her worries about how it would be used. Describing her thoughts concretely and with great detail, we can truly see the worry that Doudna had. By illustrating her personal thoughts and worries, Doudna conveys trust to the reader, which makes him more inclined to believe and understand what she is saying. She tells the audience that a scientist's comfort zone is in their lab, researching the processes of life, not going to meetings around the world to discuss the implications of these processes and their discoveries, as she decides to do after discovering CRISPR, since the technology became extremely popular after various people realized the vast potential it had for people's health and for financial profit. Describing these meetings and showing us, her audience, her reader, how she had to step out of her comfort zone to make sure that the discussion about CRISPR and its implementations would be held, illustrates Doudna in a vulnerable light, and at the same time, this honesty enables us to trust her more, to be able to relate with her worries, since we have all done something and wondered if it had been the right thing to do. We have all stepped out of our comfort zone, just as Doudna is doing with her discovery of CRISPR.